and welcome. I am absolutely delighted to have you join me today as we explore one of the most fascinating and essential mysteries of modern medicine: how our bodies transform drugs. You might be surprised to learn just how much goes on under the surface every time you swallow a pill or get a prescription from your doctor. Today, we are going to peel back the curtain on the science of drug biotransformation. In other words, what actually happens when a foreign substance, what scientists call a xenobiotic, enters your body. How does your body decide to change, neutralize, or sometimes even activate that chemical? And just as importantly, why do some drugs help while others can become toxic or stop working altogether? By the end of our time together, I promise you'll see medications in a whole new light, and perhaps understand better why the same drug might work differently for you than it does for your friend or neighbor. Let's dive in. To begin, let's talk about why this process called biotransformation really matters. Most drugs are designed to be lipophilic. That simply means they can dissolve in fat. This fat-loving quality helps the drug slip through cell membranes and get to where it needs to be. But there's a catch. Our organs that remove waste, like the kidneys and liver, find it hard to get rid of molecules that love fat. Instead, these waste removal systems prefer polar or water-soluble substances. So, in order to remove drugs from our bodies, we need to convert them from fat-loving to water-loving forms. Here's where biotransformation comes in. Think of it as the body's high-tech chemical laundry service. It tags, scrubs, and changes drugs to make them easier to get rid of. Most often, this process lowers the activity of a drug, basically inactivating it and helping our bodies to safely eliminate it. But as with all things in biology, there are a few important exceptions. Sometimes, these transformations do not just inactivate drugs. They can create new, active forms, sometimes even making a drug work for the first time. Codeine, for example, is converted into morphine in your body, which is actually what gives it its pain-relieving power. Other times, the transformation can backfire, producing toxic or harmful byproducts. A classic example here is acetaminophen, the common pain reliever. In rare instances, its metabolism can create a highly toxic substance that can damage the liver. Where does all this amazing work happen? The primary site is the liver, known for its central role as the chemical processing plant of the body. But it is not alone. Cells in your intestines, kidneys, lungs, skin, brain, and blood all contribute to this process. Now let's walk through how biotransformation actually unfolds. It happens in two main stages: Phase 1 and Phase 2. Imagine Phase 1 as the preparation phase, and Phase 2 as the finishing or packaging phase. Phase 1 is called functionalization. Think of it as adding a handle to a suitcase to make it easier to carry. In this phase, the body introduces or exposes little groups of atoms, like OH, which stands for hydroxyl, NH2, an amino group, SH, a sulfur group, or COOH, a carboxyl group, onto the drug molecule. This is done through reactions like oxidation, reduction, or hydrolysis, which are just chemical ways of saying the body tweaks or breaks bonds in the drug. The heavy lifters here are a family of enzymes known as cytochrome P450, often just called CYP. They are master chemists, precisely modifying drugs so that they're ready for the next step. Alongside CYP, other helpers include enzymes like flavin monooxygenase, alcohol and aldehyde dehydrogenases, esterases, amidases, and epoxide hydrolase. Think of them as the supporting cast in your body's chemistry show. Then comes Phase 2, the conjugation stage. Here, the body couples the now prepared drug from Phase 1, or sometimes even the original drug itself, with a small, natural molecule from the body. It is almost as if the drug is being neatly wrapped up for shipping. The result is a new compound that is highly polar, meaning it dissolves easily in water and can be efficiently removed. Several special enzymes coordinate this step, each tailoring a different wrapping paper. For example, glucuronidation uses the enzyme UGT to attach a sugar-like molecule, making it water-friendly. Sulfation uses salt enzymes to add a sulfate group. Acetylation, methylation, glutathione conjugation, and amino acid conjugation. All of these pathways make drugs safer and easier to excrete. It is worth noting that some drugs skip straight to Phase 2, like isoniazid, without any Phase 1 modification. Now let's zoom in on the star of Phase 1, the cytochrome P450 system, or CYP for short. If your body's drug metabolism was an orchestra, the CYP enzymes would be the conductors, directing a complex symphony of chemical reactions. The CYP system is actually a superfamily of proteins that contain heme, a special molecule that helps shuttle electrons. Most are found in the smooth endoplasmic reticulum. Think of it as your liver's chemical factory. They rely on oxygen and another protein partner, cytochrome P450 reductase, to do their work. Each CYP enzyme is labeled using a code. For example, CYP2D6 means family 2, subfamily D, gene number 6. Among all the CYPs, a handful handle the vast majority of drug metabolism. The most important are CYP3A4 and 3A5, which together process about half of all medications. Others, CYP2D6, 2C9, 2C19, 1A2, and 2E1, are also major players. These enzymes, however, are highly sensitive to outside influences. Some substances can induce or speed up their activity. This happens when certain drugs, foods, or chemicals flip genetic switches. For instance, the antibiotic rifampicin, the seizure medication phenytoin, the herbal supplement St. John's wort, and even charcoal-broiled foods can all trigger more CYP activity. On the other side, some molecules inhibit or slow CYP enzymes, either by competing for the same spot or permanently disabling them. Azole antifungal drugs, some antibiotics called macrolides, and even compounds found in grapefruit juice can all act as inhibitors. The result? Drugs stick around longer and can build up to unsafe levels, especially those with a narrow window between safe and toxic, like warfarin, theophylline, cyclosporine, and HIV protease inhibitors. You are probably beginning to see that our ability to handle drugs is not the same for everyone. One big reason lies in our genes, a field known as pharmacogenetics. Let's explore a few common genetic variations that shape drug transformation. We call these polymorphisms, which just means differences in the DNA instructions for making these metabolic enzymes. Take CYP2D6. About 3 to 10% of people of Caucasian descent are what we call poor metabolizers. Their bodies cannot break down drugs, like some blood pressure medications or tamoxifen, very well. So they may experience side effects, or the drug might not work at all. Meanwhile, groups such as Ethiopians and Saudis can have extra copies of this gene, making them ultra-rapid metabolizers. For them, certain drugs like codeine can be dangerously strong, or antidepressants might not reach effective levels. Other genetic examples include people in Asia who are poor metabolizers for CYP2C19, especially when processing anti-seizure medicines. Some individuals have CYP2C9 variations, which slow the removal of drugs like warfarin, raising the risk of bleeding. There are many more. Different genes affect how people process nicotine, heart medicines, painkillers, and more. Phase 2 enzymes vary as well. Some people lack cholinesterase, leading to prolonged muscle relaxation after anesthesia. Slow acetylases, who make up about half of whites and blacks, may develop nerve issues with isoniazid or lupus with certain drugs. There are even rare changes that can make simple painkillers, immune-suppressing drugs, or chemotherapy especially dangerous for some. But it is not only genes that shape our drug responses. Everyday factors make a big difference too. First, consider your diet and environment. Vegetables like broccoli or Brussels sprouts, and even grilled foods, can boost specific CYP enzymes. Grapefruit juice can block certain enzymes in the intestine, raising the levels and risks of some drugs. Pesticides in the environment also play a role. Age matters. Newborns start with low metabolism capacity, and the same goes for the elderly, who have lower liver blood flow and enzyme activity. Even gender has a small effect. Male rats metabolize some drugs faster, though the difference is less striking in humans. Illness is also critical. Liver disease, heart problems, lung cancer, thyroid disease, infections, and even cancer can all slow down enzyme activity and drug clearance. This means drugs can last longer than expected and build up to toxic levels. Now let's talk about what happens when drugs and foods interact. When one drug causes your body to crank up its enzymes, that is called enzyme induction. This can be problematic. For example, the antibiotic rifampicin can make oral contraceptives less effective. On the other hand, Some drugs inhibit the enzymes that break down other drugs, leading to toxicity. A well-known example is combining certain antifungal drugs with heart medications, which can trigger dangerous heart rhythms. Grapefruit juice can dramatically raise the level of some drugs by shutting down intestinal enzymes that would otherwise remove part of the medication. And sometimes drugs compete for the same resources needed for conjugation, like glutathione, which can make toxicity worse. Another key concept is whether a drug's removal depends on blood flow, what we call flow limited, or on how much enzyme is present, what we call capacity limited. High extraction or flow limited drugs are mostly cleared as they pass through the liver. If the heart is failing and less blood gets to the liver, these drugs can hang around much longer. On the flip side, low extraction drugs rely on the enzyme's speed. They are very sensitive to inducers and inhibitors. Here is where the field becomes truly promising: personalizing therapy using genetic information. While genetic tests can help guide prescribing for drugs like warfarin, anti-cancer medicines, or clopidogrel, these tests are not standard for every medication yet. Cost, logistics, and the need to train clinicians are still hurdles, but the potential benefits are enormous. Safer drugs, fewer side effects, and potentially saving over 100,000 lives each year from avoidable reactions. So let's distill these big ideas into a few key takeaways. First, your body is wonderfully clever at transforming and eliminating drugs, relying on a two-phase system, preparation and wrapping, to make medicines safe and removable. Second, a handful of enzymes, especially the CYP family, handle most drug transformations, but their activity can be dialed up or down by genes, diet, age, health conditions, or other medicines. Third, who you are, your genetic makeup, can completely change how you respond to medication. That is why personalizing therapy and being aware of possible interactions is so important. And finally, as science advances, testing for these variations and avoiding dangerous drug combinations can make treatment safer and more effective for everyone. Thank you so much for joining me on this journey through the remarkable world of drug biotransformation. I hope you've discovered just how dynamic and personal medicine can be. I would absolutely love to hear your questions, thoughts, or stories. So please do not be shy. Let's chat about it in the comments below. Until next time, take care and keep nurturing your curiosity. Goodbye.